So now that we've talked about glycolysis, the next step in cellular respiration is the citric acid cycle. This is also called the Krebs cycle, as you see here. This diagram is a little more detailed than you'll need to know, so don't worry about memorizing the names of each of those molecules. What we've covered is that in glycolysis, a cell will break glucose down into 2-3 carbon molecules, where you get a net gain of 2 ATPs, and the remaining products are called pyruvates from glycolysis. And this picks up, of course, where glycolysis left off. So after glycolysis, we're left with a 3-carbon molecule chain called pyruvic acid, or in other books they call it pyruvate. That's okay, that's beside the point. Essentially, one step needs to occur before these molecules can enter the citric acid cycle. What essentially happens is pyruvic acid is oxidized, and in that process it loses one of its carbon molecules, and one molecule of carbon dioxide is then generated as a waste product. So that makes pyruvic acid something else entirely, with only 2 carbon molecules. It becomes acetic acid. Then NADH comes in, accepts 2 electrons, or takes 2 electrons from this acetic acid, and coenzyme A is present, which then binds to the acetic acid molecule. This then is what we call acetyl-CoA. Your book describes acetyl-CoA as the escort to get these 2 carbon molecules into the mitochondrion to start the citric acid cycle. I have read elsewhere that this oxidation process actually already occurs within the mitochondrion, that somehow that pyruvic acid does make it into the matrix and then it's oxidized, but your book states it a little differently. I won't test you on that specific part, but just know that this is the intermediate step after glycolysis, before the citric acid cycle, where pyruvic acid is oxidized into acetyl-CoA, and that has to happen before it can go into the citric acid cycle. This is a reminder that glycolysis occurs in the cytoplasm of the cell. The citric acid cycle occurs within the matrix of the mitochondrion. And then your electron transport chain, the proteins that actually perform that process are embedded in the inner membrane here. So right now, what we're going to talk about is happening inside of the matrix of the mitochondrion. What happens here, acetyl-CoA becomes oxidized essentially in the citric acid cycle, and it goes through 9 different reactions as this molecule cycles through. There are really 9 different steps which lead to 2 carbon dioxide waste products, an ATP, 3 NADHs, and FADHs. So we're going to talk a little bit more about what exactly happens during this cycle. Acetic acid enters as acetyl-CoA and gets attached to another molecule that has 4 carbons on it. That then becomes citric acid, for which this process is named. One thing you want to keep in mind is that this whole process is designed to transfer electrons, the original electrons from your food molecule, using several different steps in order to gain that energy and put it onto an ATP so that it can be used later. Even though there are 9 actual steps in which this happens, your book basically breaks it down into 6. First, your 2-carbon acetic acid joins the 4-carbon molecule here to make citric acid. Then there are actually 2 individual steps which each take 1 carbon off of this citric acid molecule. And so that's step 1, step 2, and that's how you get your 2 carbon dioxide molecules as waste products. As the cycle continues, there is a step right about, say here, that includes succinyl-CoA. At the succinyl-CoA, right about here, is when ATP is generated. Now remember that 2 acetic acid molecules will come from 1 glucose molecule, so this whole cycle can happen twice for each single glucose, meaning that one cycle of citric acid generates 1 ATP, but for each glucose molecule, you will get 2 ATPs out of the citric acid cycle. I hope that makes sense. The other important step that comes from the citric acid cycle is the generation of 3 NADHs and FADH2. We've already seen NADH. FADH is just a similar molecule with a little bit different shape that is used as an electron acceptor. Even though ATP is the ultimate goal, in this step, NADH and FADH are critically important because what they do is they carry those electrons to the next process in cellular respiration, which is the electron transport chain. Now, just to bring things a little bit closer, we're going to talk about the inputs and the outputs of the citric acid cycle here. So remember that for every 1 acetic acid, 2 carbons that comes into the cycle, which will also use an ADP and a phosphate, 3 NADHs, and 1 FAD. You will then get 2 carbon dioxide molecules as waste. 1 ATP is generated for every single turn of the citric acid cycle, which will turn twice for every 1 molecule of glucose. So ATP, 3 NADHs, and 1 FADH. And again, these are important. These are electron holders that bring those electrons to what we're going to talk about next, which is the electron transport chain, and that will be in a different video.